Imagine an exploration mission through a strange and hostile environment. Now, place yourself inside the imaginary vehicle, the 1/10 of a millimeter long Stromatolite Explorer. The mission is to probe a microbial mat, a complex and very ancient assemblage of microorganisms less than 5 millimeters thick. Automated transponders will take chemical readings every 1/4 of a millimeter as the vehicle descends. Diving into a living mat is like going back in time to explore some of the earliest forms of life on Earth. Microbial mats have been around for over 3 billion years, making them Earth's earliest biological communities. Finding the remains of microbial mats on other planets would be powerful evidence that life once thrived there. That's why understanding microbial mats is of such interest to NASA. In the uppermost region of the mat, diatoms reign supreme. These single cells are encased in houses made of glass. Diatoms convert sunlight and carbon dioxide into fuel molecules and oxygen through the process of photosynthesis. Now the explorer and crew enter the realm of filamentous cyanobacteria. Each strand is a chain of single-celled prokaryotes. No nucleus in these cells, just a long strand of DNA.
Depth 0.5 millimeters. Oxygen 600 micromolar. Hydrogen sulfide negligible. pH 9.7.
Like other solar-powered photosynthesizers, a cyanobacterium produces dissolved oxygen as a byproduct of photosynthesis. Here we see how Earth's atmospheric oxygen originated, bubble by bubble.
Depth 2.6 millimeters and descending. Oxygen negligible. Sulfide 75 micromolar. pH level 7.2.
Because the only light that gets down to the deeper part of the mat is in the form of red to near-infrared radiation, the bacteria here, purple sulfur bacteria, use that radiation and the hydrogen sulfide present to perform a kind of photosynthesis that doesn't produce oxygen, an oxygenic photosynthesis.
Depth 4.3 millimeters. Hydrogen sulfide 125 micromolar. Oxygen 0.
The mat, though relatively thin, shows a steep chemistry gradient from top to bottom. The explorer has descended through layers composed predominantly of diatoms, then cyanobacteria, of purple sulfur bacteria, and finally, sulfate-reducing bacteria. Oxygen from photosynthesis at the surface of the mat never makes it to the bottom. So the bacteria down here have evolved to use sulfate to break down or burn organic matter.
Alert. Reserve power at critical level. Insufficient solar radiation for successful recharge. Warning.
The mat community contains filamentous bacteria that move between the sulfur-rich deeper layers and the oxygen-producing upper layers in a daily cycle. If the crew can remember this, they might have a chance to hitch a free ride back to the surface. When the sun goes down, the chemical output of the mat community adjusts for life in the dark. A peek at the updated transponder data reveals a new set of readings. A microbial mat is a community that changes dramatically in the first few millimeters. Descending, the mat changes from a photosynthetic, oxygen-producing layer to a murky realm where sulfur-munching bacteria prevail. The activities of these simple life forms create an alkali environment where minerals accumulate and, over time, a stromatolite will form, one of the telltale signs to look for in our search for life on other worlds. Someday, not so far away, when an exploration vehicle from Earth begins probing the soil of a planet such as Mars, it will carry instruments for analyzing biosignatures, minerals, and organic matter that may have been left behind by mats that once grew on the shores of salty Martian seas.